This protocol describes a method for comprehensive, unbiased molecular characterization of a single sample using both mass spectrometry, using metabolomics, proteomics, and lipidomics, and nuclear magnetic resonance spectroscopy platforms. The significance of this approach is that we can characterize a biological system downstream of the genome through identifying different types of molecules, allowing us to infer metabolic and decomposition pathways.
The sequential extraction technique allows extraction of bioavailable polar compounds using water, followed by MPlex to capture any additional polar compounds or metabolites, as well as proteins and lipids from a single sample.
Visual demonstration of this method is critical because it guides the scientist through separating the 3 layers during the second extraction step and splitting the extracts between different analysis instruments.
Integrative multi-omic analyses empowers more effective investigations and complete understanding of complex biological systems. This robust method extracts a wide diversity of molecules and is applicable to diverse sample types.
Begin this procedure with collection and preparation of the samples as described in the text protocol. Using an ethanol-washed stainless steel utensil, aliquot 50 milligrams of each of the dried samples into individual 2 milliliter glass tubes. Add 1 milliliter of distilled, degassed water to each sample. Then, cap the vials and shake for 2 hours on a shaker table. Centrifuge the samples at 15,000 times gravity for 30 minutes, and then allow the solutions to stand at room temperature for 20 minutes. Decant and save the supernatant from each sample.